My name is John Kellie, and I am an Associate Fellow and Investigator in the Bioanalysis, Immunogenicity, and Biomarkers group at GlaxoSmithKline. My role is centered around large molecule mass spectrometry and measuring and analyzing large molecules and antibodies for preclinical and clinical studies. So I think some of the biggest challenges that our group is facing currently is around some of the complex biotherapeutic molecules such as fusion proteins and ADCs. And how does our group deliver the most high quality data that really enables them to make informed decisions, these preclinical and clinical teams? And I think then we can think about the different types of measurements that are routine in our laboratory. And we think about ligand binding assays, and we think about trypsin digestion-based LC-MS assays. But if we can provide more information from the mass spectrometry space, then our goal should be to utilize all the tools in our toolbox and give the best information possible. So our technologies are centered around liquid chromatography coupled to high-resolution mass spectrometry. And we often would also utilize immunocapture in the front end purification standpoint. So we can purify the molecule of interest away from the matrix or plasma proteins, for example. And then we can provide another stage of separation, purification through the LC standpoint. And then for the mass spectrometer, using the mass spectrometer, we can measure a full M over Z range that enables us to get a complete picture of the molecular mass and masses of the protein molecules. And when we do this, then we get a rich layer of information about possible biotransformation, possible metabolism of these protein molecules. So some of the advantages that we've seen is when you can monitor the intact mass of a biotherapeutic, when you can monitor the whole molecule at the subunit level and at the intact mass level, you can really learn more about the molecular integrity of the molecule. So what happens to the molecule after it's dosed is a very important question, and that may be complementary information that you just don't get from a traditional ligand binding or trypsin digestion assay. So not that the other assays wouldn't and shouldn't be used, but we can add additional information about intact and subunit level mass measurement that other approaches just can't quite capture. And we can utilize that then to better understand what happens to the molecule and make informed decisions based on that. So for large molecule bioanalysis, when we measure the molecular masses by mass spectrometry, it can really enable good decision-making power for drug development and drug delivery. We can think about how these mass measurements support discovery phase, drug development. We can think about how it supports clinical phase drug development through critical quality attribute monitoring from clinical samples. And so we can really use the information from the mass at the mass level, so an intact mass or some biotransformation product that's representative of, let's say, amino acid clipping. And we can better understand the product and the impact on delivering that to patients. So in 5 to 10 years, I hope that antibody and protein monitoring from in-life samples is more routine at the large mass level. I certainly hope that as high-resolution mass spectrometry continues to grow in popularity and those instruments are more prevalent in laboratories, that researchers are not afraid to try and tackle the next big step, to move away from a trypsin digest and move towards an intact mass measurement for routine PK concentration, or to, let's say, move away from a traditional ligand binding assay as the sole supporter and utilize a ligand binding assay and also use subunit mass monitoring. And so we can begin to also think then, as these methods grow, how this fits into the regulated space. And we can begin to think today about how to handle the data, how to handle the data processing, so that it's on the radar of regulators, and we can begin to have discussions so that when the time comes to utilize an intact antibody assay to support a regulated study, the regulators already understand the data and they're already on board.